Welcome back. We have come now to the end of our lecture on synaptic transmission, and we've come really to the end of our whole unit here on neuroscience fundamentals. I've had a really good time making these videos for you. I've had a good time interacting with you guys in our one-on-one sessions, and I'm looking forward to doing it one more time on Monday. So, in our previous videos, you'll remember that we've covered the basics of synaptic structure and function, and in particular we focused on these methods, the mechanisms of how vesicles fuse to the membrane and release their neurotransmitters. We also talked about several different kinds of neurotransmitters, and we looked in detail at the neurotransmitters here in this table. And today we talk about the part 3 of this diagram here, sort of the last part of the equation, which is the receptors, the place where the neurotransmitters bind, and that really cause the effects that those neurotransmitters have. So we're going to start off talking about receptors. We're going to take what we know then about the receptors and use it to revisit our understanding of EPSPs and IPSPs, and then we're going to talk about selected neurotransmitter systems and their relationship to behavior. And you'll see for this video, I've chosen this image here. Hopefully you recognize this individual as the athlete and civil rights and anti-war activist Muhammad Ali. And the picture on the left here is when he was a young man, when he was, at the peak of his abilities as an athlete. And on the right here, he is 40 years later suffering from Parkinson's disease. And I'm using this as an illustration of our topic today because we know that the symptoms of Parkinson's disease are caused by the loss of neurons that produce the neurotransmitter dopamine. And it's, in my mind, this is a very stark example of just how one neurotransmitter system can have profound effects on behavior, such that if you lose the neurons that produce that neurotransmitter, it can be very, very devastating for your health and for your well-being. Yes, some of you may know, Parkinson's is a neurodegenerative disease, and it's a disease that's still with us. There are some treatments available for Parkinson's disease, but we still don't understand what causes it, and we still don't understand what the cure is. One of the, this is, I would say, is another one of the great challenges in neuroscience, is understanding the relationship between neurotransmitter systems and behavior, and how we can change the course of diseases that involve these neurotransmitter systems, like Parkinson's. Okay, so remember, this is our synapse here. We have action potentials entering the axon terminal. We have the vesicles binding to the presynaptic membrane, and we have a neurotransmitter being released into the synaptic cleft, and those neurotransmitters are then going to bind to the receptors and are going to activate those receptors. And the first sort of key idea that you need to understand about neurotransmitters and receptors is that they are specific to one another. They are keyed to one another, meaning that dopamine is only really going to bind to and is really going to only activate dopamine receptors. And likewise, dopamine receptors are really going to be bound by and are really only going to be activated by dopamine. So even if you have other neurotransmitters, let's say, present in the synapse, it's only going to be the specific neurotransmitter that is going to bind to its specific receptor. They are keyed to one another. The sort of second important idea here is the idea that whatever effect we see from the release of neurotransmitters at the synapse, that release is going to be determined by that receptor. So, you might hear me say, or you might have heard me say things like, this neurotransmitter is excitatory, or this neurotransmitter is inhibitory, or this other neurotransmitter has a modulatory action. But that's actually a little bit imprecise. What we really mean is that those neurotransmitters, when they bind to their specific receptors, those receptors are going to have a excitatory action or inhibitory action or a modulatory action on this postsynaptic cell. And so again, when we think of, what is the function of the synapse? It's to translate electrical signals into chemical signals and then back into electrical signals. And it's really the receptors that are responsible for that last part, going from the chemical back to the electrical. Now, in very broad terms, we can categorize these receptors as being either of one of two kinds, either ionotropic or metabotropic. And I just want to emphasize these are broad divisions. There are many, many different kinds of receptors, but very generally we can divide them into ionotropic and metabotropic. Now, some neurotransmitters, the only receptors that they will bind to are metabotropic receptors. Other neurotransmitters may only have ionotropic receptors to bind to, and in some cases there are neurotransmitters that will bind to both a metabotropic kind of receptor and an ionotropic kind of receptor. And I'm going to give you some examples of all three of those cases. First off, though, we want to define what ionotropic and metabotropic are. Well, as it turns out, I've actually already told you about ionotropic receptors. So this is the point I was making before. So you'll remember when I talked about these amino acid neurotransmitters, glutamate, GABA, and glycine, and I said that glutamate is an excitatory neurotransmitter. What I mean by that, again, as I said before, is that glutamate tends to bind to receptors that have excitatory actions in the postsynaptic cell. All right. So you remember, I said to you before that you already have been introduced to ionotropic receptors, but just under a different name. Before, when I introduced them to you, I called them neurotransmitter-gated ion channels. And what we see here, again, these are channels that are in the postsynaptic membrane that when a neurotransmitter binds to them, they open and they let ions either into the cell or out of the cell, and they do so with specificity, meaning that they're only going to let some ions through but not others. Ionotropic receptors, they're just neurotransmitter-gated ion channels that open when a neurotransmitter binds. And as you can expect, right, what happens when we open a channel to let ions into or out of the cell? Well, remember that's going to change the voltage inside the cell. If we're letting positive ions out or if we're letting negative ions in, then that's going to decrease the voltage of the cell. If we let the positive ions in, that's going to increase the voltage of the cell. So here in the silly little table are just sort of examples of some neurotransmitters that you've heard about before. The receptors that they bind to, the ionotropic receptors that they can bind to, the ions that those receptors are specific for, and the effects that they have. So I just mentioned glutamate. In the central nervous system, when glutamate is released, the ionotropic receptors that it will bind to are called AMPA receptors. AMPA receptors, as it turns out, are neurotransmitter-gated ion channels, and when glutamate binds to the AMPA receptors, they open, and they open and allow sodium into the cell. And hopefully by now you should know what happens when we let sodium into the cell. We're going to get a depolarization. We're going to increase the membrane potential, excite that cell, get it closer to firing. Likewise, for the neurotransmitter GABA, I said before that this is an inhibitory neurotransmitter. Well, what I mean is that in many synapses in the central nervous system, GABA will bind to the GABA A subtype receptor, which is an ionotropic receptor. And if we look at what that GABA A receptor does, what we see is that it is an ion channel that binds GABA, and when it opens, it allows chloride to pass. And remember, there's more chloride on the outside of the cell, so chloride will enter the cell, and you should be able to answer what happens when we let a negatively charged ion into the cell. We're going to decrease the membrane voltage in that cell. We're going to hyperpolarize that cell. We're going to get it farther away from an action potential. And finally, our final example here is in the peripheral nervous system. If remember, the neurotransmitter acetylcholine has a role at the neuromuscular junction. That role is mediated by what's called the nicotinic subtype of the acetylcholine receptor. That's the receptor that is on the muscle cells that are there at the neuromuscular junction. When acetylcholine binds to nicotinic receptors, it opens up, and this is an example of a channel that is semi-specific. It lets both sodium and potassium through. Even though those may seem to have opposing effects, the net effect of opening these nicotinic receptors is depolarization. And remember, in muscle cells, that depolarization we call the end plate potential. All right, so those are ionotropic receptors. Now we're going to talk about the second kind of receptors, which are metabotropic receptors. And unlike the ionotropic receptors, the metabotropic receptors usually are not channels. They're made out of proteins, and they live inside of the membrane. But what they do, what you can think of these as, is as the starting point for protein-protein enzymatic reactions that will have indirect effects on the cell membrane. And for this reason, because they have indirect effects, very often we say that the effects of metabotropic receptors are modulatory, meaning they're not sort of directly acting on the cell, they modulate something else that's going on in the cell. You'll see the book gives an example of one kind, probably the most common kind of metabotropic receptor, which is called a G protein coupled receptor. And what the heck is a G protein? So in general, in a G protein coupled receptor, you have one portion of the receptor that spans the membrane. The extracellular side of this receptor is going to contain the binding site for the neurotransmitter. So that's where the neurotransmitter is going to bind. And then on the intracellular side of the receptor, you're going to have this G protein complex. It's going to be a group of 2 or 3 or 4 proteins called G proteins. And what I'm going to show you in just a second are the effects of these G proteins. They basically are signaling molecules that are going to be able to send messages throughout the cell. Now, the book kind of gives two basic examples of what happens when you activate a G protein coupled receptor. And we'll start with the example on the left, and then we'll talk about the example on the right. So for some of these G protein coupled receptors, when neurotransmitter binds, we activate this G protein complex. Technically what happens is usually there's some sort of enzyme cascade that results in the phosphorylation of one of these G protein subunits. What then very frequently happens is that this phosphorylated G protein subunit detaches from the receptor and enters the extracellular fluid and becomes a messenger protein. And in some cases, that messenger protein can go to a ion channel that is located within the cell membrane and can activate that ion channel and either open that ion channel or close that ion channel or modulate its function in some other way. And by doing that, by acting on these ion channels, can have a sort of, second order effect on the membrane potential in that cell. This is just one example of what these G protein coupled receptors can do. And again, just to sort of emphasize here that this is this is an indirect effect. It's not the receptor that is directly changing the voltage inside the cell, that is directly changing ion flow. It's this G protein, which becomes a signaling molecule that can then go and modulate the activity of an ion channel and therefore modulate the voltage inside the cell. The second example that the book gives, and I think this is actually probably one of the most common examples, or sort of one of the most common effects of activating metabotropic receptors, is one where we bind the neurotransmitter, we activate our G protein complex, and we release our G protein subunit. But instead of going and acting directly on an ion channel, that subunit will activate or phosphorylate yet another enzyme that lives within the cell. and the activation of that enzyme, so hopefully you remember this from your basic, molecular biology, can initiate a cascade, a sequence of second messenger protein-protein interactions. And here, we can think of many, many different effects that this could have. So, for example, it could be you could imagine an instance where the activation of our G protein subunit activates a cascade of second messengers, which eventually translocate to the nucleus and stimulates the production of more neurotransmitters that will eventually be manufactured and placed inside the cell membrane. Right. So and when that happens, we can imagine that the effects of this receptor are going to eventually have an effect on the membrane voltage inside the cell. But of course, it's going to be happening at a much slower time scale and in a much more indirect fashion, sort of through these intracellular second messengers. Now, these are just two examples of how activation of metabotropic receptors can change the activity or can modulate the activity of neurons. There are many, many more examples of this, way too many to go through, obviously, in an introductory course. But the main idea that I want you to get here is that sort of once you're into the realm of metabotropic receptors, you're talking about receptors that can modulate or change sort of nearly any aspect of how that neuron functions and how it responds to its incoming inputs. And finally, here's a table here with just some examples of metabotropic receptors. And first thing I actually want to point out is the first two here, acetylcholine and glutamate. In the table I showed you just a few moments ago, the first two entries were also acetylcholine and glutamate. So these two neurotransmitters, these are neurotransmitters for which there are both ionotropic receptors and metabotropic receptors, right? So this is an example where we have the same neurotransmitter, but its effects are going to be different depending on what kind of receptors it activates. Now, looking at acetylcholine, as it turns out, its metabotropic receptors are called muscarinic subtype receptors. And in both the central and peripheral nervous system, there are actually 5 different kinds of muscarinic receptors. And each one of these 5 different kinds of receptors are going to have subtly different effects through different kinds of second messenger actions in the manner that I just described. One example here is if we go back to our example of the frog heart experiment and the stimulation of the vagus nerve. Remember, we identified that it was acetylcholine that's being released there, and as it turns out, the receptor in cardiac muscle that is receiving that acetylcholine signal is the M2 subtype receptor, and we can attribute the slowing of the heartbeat to the second messenger effects of the M2 receptor subtype. Glutamate is, again, another neurotransmitter that we've talked about before that has both ionotropic and, as it turns out, 8 different subtypes of metabotropic glutamate receptors. These are usually just called mGluR1 through mGluR8. And if we just look at an example of what these do, both mGluR1 and mGluR5, when they are activated, the sequence of protein-protein interactions eventually results in the release of intracellular stores of calcium into the cytosol. Here, these are metabotropic glutamate receptors whose second messenger effects include calcium signaling inside the cell. Finally, for dopamine has 5 different major subtypes of receptor labeled D1 through D5. And D1 receptors, as it turns out, just as an example, have effects on some of the classical intracellular second messengers. So, hopefully you remember from your molecular biology, the second messenger is adenylyl cyclase, cyclic AMP, and protein kinase A, PKA. Right? D1 receptors directly activate that sequence of second messengers. All right. That just kind of gives you a basic overview of what receptors are and how they have their basic actions. And now that we know about these, and now that we know in particular about ionotropic receptors, I just want to go back and revisit a topic that we talked about before, which was EPSPs and IPSPs. And right before, I just talked about, the idea that there were excitatory pathways where if you stimulate them, you get this increase in membrane voltage, this depolarization that gets us closer to threshold, closer to an action potential. And if you stimulate an inhibitory pathway, we get this decrease in membrane potential, this hyperpolarization that gets us farther away from an action potential. But now we can sort of be more specific about what's happening here. These excitatory pathways, these are glutamatergic synapses. And we know that when we're stimulating these glutamatergic synapses, the glutamate is crossing the synapse, it's binding to AMPA receptors. And those AMPA receptors, we know, when they're activated, these are ionotropic receptors. They open and they allow sodium into the cell, which of course depolarizes the cell. So EPSPs are really the result of the activation of glutamatergic synapses. Likewise, these IPSPs, we now know that what we're doing when we're stimulating these is we're releasing GABA. GABA is binding to the GABA A receptor subtype, an ionotropic receptor that allows chloride ions into the cell. And when we let negative ions into the cell, we hyperpolarize that cell, getting it farther away from an action potential. So, going back to this idea that, whether or not a cell fires an action potential depends upon the voltage here at the axon hillock, and the voltage at the axon hillock is dependent upon the summation of EPSPs and IPSPs coming in all throughout the dendritic tree of the cell. Another way of putting this is saying that whether or not a cell fires an action potential depends upon the balance of the activity at glutamatergic synapses, which are of course causing EPSPs and promoting excitation, along with the GABAergic synapses causing IPSPs and promoting inhibition. Now we can say, what are EPSPs and IPSPs? They are the result of the activation of glutamatergic and GABAergic synapses. We can finally now also talk about this idea of graded transmission. Now, remember, we talked about the main function of the synapse, which is the transformation of an electrical signal into a chemical signal back into an electrical signal. And we also said before in the previous lecture that action potentials are all or none phenomena. But one important thing to realize is that even though action potentials are all or none, neural transmission at a synapse can still be graded, meaning it can still fall along a spectrum. And what I mean by that is this, is that if we have at a axon terminal, if we have at a synapse, only a few action potentials coming in at a slow rate, what that's going to result in is a small release of neurotransmitter. As we increase the number of action potentials or the rate of action potentials that are incoming into the synaptic terminal, we're going to increase the amount of neurotransmitter that's released. Now, if we increase the amount of neurotransmitter that's released, that means, up to some limit, we are going to increase the number of receptors that are activated, which means that we are going to amplify whatever effect those neurotransmitters have. So if we're talking about the case of an ionotropic receptor, remember this is a receptor where we're opening a channel, we're directly modulating the membrane voltage in that dendrite. If we only have a small release of neurotransmitter, then we're only going to get a small change in the postsynaptic potential. If we have a large increase in neurotransmitter, then we're going to get a large change in the postsynaptic voltage. Now, this is sort of what this would look like at just one synapse. If we imagine now that the synaptic inputs now coming in at many different synapses all across the cell, if there are lots and lots of strong, if there are only a few inputs or weak inputs, then we're we may not spike at all, or we might only get a few spikes. However, if we have lots of neurotransmitter release all throughout the dendritic tree of a cell, then we're going to fire more, and so on and so forth. So this is an important idea to keep in mind. Even though an individual action potential by itself is an all or none phenomenon, in actual sort of real neurons, there's usually more than one action potential happening at once, and it's the rate of action potentials that is going to determine how much neurotransmitter is going to release, and that's going to determine the strength of the postsynaptic signal. And this is a way in which we can have this transformation, this electrical transformation that maintains the intensity of the signal. Okay, so to finish up, we're going to take sort of a little bit of a step back, and we're going to talk now about a few different neurotransmitter systems and their relationship with behavior. We've talked in great detail about the mechanisms and the detailed mechanics of neurotransmission. Now what I want to do is just talk a little bit about how these neurotransmitter systems relate to behavior. And this actually, it turns out, is a very, very difficult topic. I gave you this example of the role of dopamine in Parkinson's disease. But as it turns out, it is impossible to look at one specific behavior and to attribute that behavior to just one neurotransmitter system or one receptor subtype alone. Even the most simple of our behaviors are usually going to result from activity distributed over many different circuits and using many different neurotransmitter systems. In this next section, I just want to emphasize that what we're going to talk about here are very broad and general associations between some neurotransmitter systems and certain classes of behavior, as well as some illnesses that have a cognitive or behavioral component. But I don't want you to get the impression that there's a one-to-one relationship between certain neurotransmitter systems and certain kinds of behaviors. What we're going to be talking about here are broad associations, things that we have basically learned through research and through clinical studies. And we're going to cover today what are sometimes called the four major activating systems within the brain. Activating systems is kind of an old-fashioned term. It basically means neurotransmitter systems that in general are involved in generating behavior, in sort of getting us into states of arousal or wakefulness or activating our behaviors. And the four neurotransmitter systems we're going to talk about here are acetylcholine, serotonin, dopamine, and norepinephrine. They all share a couple of common features. The first thing that these four neurotransmitter systems share is that they originate with really a fairly small number of neurons. And what I mean by that is compared to sort of all of the neurons throughout the brain, the number of neurons that produce and release acetylcholine or serotonin or dopamine or norepinephrine make up a very small number of neurons relative to all of the neurons in the brain. And most of these are going to be located in the brainstem. However, and I've mentioned this before when we talked about neurotransmitters, even though it's only a small number of neurons that synthesize and release these neurotransmitters, the neurons that do release these neurotransmitters tend to send their axons widely throughout the brain, throughout the cortex, the basal ganglia, the thalamus and hypothalamus. And because of that, they are able to affect and modulate the neurotransmission throughout many different parts of the brain. Most of these, at least within the central nervous system, have only modulatory function. And again, what that means is they're not directly exciting or directly inhibiting neurons, but they're changing the responses of those neurons somehow through second messenger systems. As I mentioned before, they're involved in many, many different kinds of behaviors, and they're also involved in different mental or psychiatric disorders, and are frequently the targets of drugs to treat those disorders. So we're going to start with acetylcholine, or what's called the cholinergic system. I think the book actually does a good job in summarizing these, so I'm just going to kind of hit the main points that the book hits. So the neurons that synthesize acetylcholine in the central nervous system are usually located in the brainstem or in the basal forebrain for the most part. It projects widely to the cortex and to the basal ganglia, as well as a few other regions. And their major functions seem to be in modulating states of wakefulness or arousal, as well as our attention, our ability to attend to specific stimuli in the world. And clinically, the loss of these neurons or the loss of cholinergic signaling is associated with Alzheimer's disease. The second major system that we're going to talk about is the dopaminergic system. The neurons that synthesize dopamine are located in the substantia nigra and in the ventral tegmental area. Those are located in the midbrain. These send very strong projections to the basal ganglia. They also project to the cortex as well and a few other regions. And dopamine actually has a number of different sort of behavioral roles. One of its big one is in the initiation and coordination of movement. However, its other one also has to do with aspects of reward, motivation, and reinforcement. And dopamine also has effects related to attention and other aspects of higher cognition. Clinically, we've already talked about the role of dopamine in Parkinson's disease. Remember, it's the loss of dopamine neurons and the loss of dopamine signaling that is responsible for many of the major systems in Parkinson's disease. And in fact, one of the sort of first and most basic treatments for Parkinson's disease is a drug that essentially enhances dopamine signaling. Another sort of major place where we find a clinical role for dopamine is in drug abuse. Basically, almost every drug of abuse, whether it be stimulant drugs, opiate drugs, inhalants, almost every major drug of abuse will one way or another increase the release of dopamine in certain basal ganglia centers. And it's that activation of dopamine in those basal ganglia centers that seems to be have a lot to do with the addictive and reinforcing properties of drugs of abuse. And we also know that dopamine has an involvement in schizophrenia, ADHD, and depression. And in fact, one of the reasons we know this is that many of the drugs that treat these conditions act, at least in part, on the dopamine system. The third system we're going to talk about is the noradrenergic system. Noradrenergic neurons, they're also located in the brainstem in a part of the brain called the locus ceruleus. They project widely to the cortex, but very interestingly, they do not project to the basal ganglia. They're one of the few neurons in these activating systems, or this is one of the few activating systems that does not activate the basal ganglia. And its functions generally seem to be having to do with general arousal, states of wakefulness or sleep, attention, but also stress and mood. And many drugs to treat depression, bipolar disorder, and ADHD will target the noradrenergic system, meaning that they target noradrenergic receptors. And finally, we have the serotonergic system. These neurons that synthesize serotonin are located in one of several raphe nuclei. I think we actually have many raphe nuclei. These are generally located within the brainstem. They also project widely to cortex and to other regions as well. These also have a role in our mood, but also wake and sleep and arousal. And serotonin is, of course, associated with major depression. Some of you may be familiar with a class of antidepressant medications called SSRIs. Those are selective serotonin reuptake inhibitors. What they basically do is they block the channels that take serotonin out of the synaptic cleft and take them back up into the presynaptic terminal, thereby increasing the strength and the duration of serotonergic signaling within serotonergic synapses. And we also know, again, mainly through clinical applications, that the serotonergic system is also involved in OCD and schizophrenia. Okay, so we've come to the end of our lecture, and for our sort of little test your knowledge section here, I'm going to put this one more silly little table up here. And I'm going to sort of challenge you guys to sort of take a moment and see how many of the squares here that you can fill in on this table, right? So we have the neurotransmitters that we've been talking a lot about here, glutamate, dopamine, and acetylcholine. You'll remember we talked about how neurotransmitters come in families. We talked about where we can find these neurotransmitters, as well as whether they have metabotropic or ionotropic receptors or both, as well as some of their major functions. I think, if you were able to fill something in for every one of these grid squares, I think that would be very impressive. But maybe take a moment now to see how many of these you can fill out. I think if we think of some of the most basic information we can recall about these neurotransmitters, we can talk about their family. You should hopefully remember that all of these are classified as small molecule neurotransmitters. And maybe those of you who really have a good memory will remember that glutamate, they're small molecule neurotransmitters, and their subfamily are the amino acid neurotransmitters. Dopamine, their subfamily is the monoamines. And acetylcholine actually is its own really special little subfamily in and of itself. Some other things that I hope are at least hopefully easy for you to recall to mind are some of the major functions. Some of these we've talked about several times. We've talked several times now about how glutamate has mainly excitatory actions through its actions at excitatory synapses, EPSPs. We've talked about several functions for acetylcholine. We've talked several times about its role in generating muscle contraction in the neuromuscular junction. And we've also talked a little bit about its function in cardiac tissue, its autonomic function, and even we just now talked about its roles for wakefulness and attention in the central nervous system. Hopefully you remembered at least one of these. And finally, remember, we've talked about a couple different roles for dopamine, most prominently its role in the initiation of movement and its role in Parkinson's disease. Now, the rest of these you might find a little bit more difficult. Maybe if you want to pause now and go back and see if there are any that you can recall. Remember glutamate is found almost nearly everywhere in the central nervous system. Dopamine, its cell bodies are only in the midbrain, but its axons go almost everywhere. And acetylcholine is one of these that is really found everywhere, both in the peripheral nervous system as well as the central nervous system as well. And finally, we remember that there are metabotropic and ionotropic receptors, and some neurons have one kind, some neurons have both. We've talked several times for acetylcholine about its receptors, and these are just examples for glutamate and dopamine. All right, that brings us to the very end. I look forward to seeing you all again one more time on Monday. In the meantime, best of luck in preparing for class, and enjoy your weekend. Bye-bye.